at the Duke University School of Medicine, North Carolina, USA. And his group is interested in understanding the programmed cell death during intracellular infections. And made important contributions to the inflammasome field by describing the role of caspase 11 in the detection of intracellular LPS and the sensing of type III secretion system by NLRC4. And also made seminal contributions to the understanding of pyroptosis, which includes the first demonstration of the role of pyroptosis to clear in vivo infections, it's so hard, and the description of the pore-induced intracellular traps in consequence of the cell death pattern. So I feel very lucky to have him today in our course, talking about my favorite cell death modality, pyroptosis, of course. Thank you, Eddie.
Thank you for inviting me to speak to you all today. Can you turn on the screen share for me?
Márcio, coloca ele como co-host, por favor.
There we go.
Yeah.
Yes.
All right. Okay, so I'm going to talk about inflammasomes and pyroptosis today. Giving you an introduction to hopefully the entire field. I'm not going to cover everything because it's a pretty deep field, but hopefully giving you an overview of many of the larger topics. And I will give a little apology. I might have more focus on some of my own work because I had more slides nicely made for those parts of it. So little disclaimer there. And also feel free to interrupt throughout the talk with questions. Sometimes I drone on a little bit too fast. So if things aren't clear, pipe up with questions, or just if you're interested, please feel free to interrupt. All right, so I'm going to tell you about inflammasomes, which are members of innate immune sensors. So in innate immunity, mammals are interested in surveilling the extracellular space, the vacuolar space, cytosol, and perhaps the nucleus. And we do this with pattern recognition receptors, the goal of which is to discriminate self from non-self. And in innate immunity, we're trying to do this innately with germ-coded receptors. So these have to be receptors that detect something that is slow to evolve in pathogens. So it is difficult for them to evolve to evade the sensor, which is very important because if we want to have a germline encoded receptor, it will be very difficult for us to change it over evolutionary time, whereas microbes can evolve much faster than that. So that has to be detecting something that's difficult for them to change. Pattern recognition receptors can detect specific molecules that are parts of the pathogen. We call these microbe or pathogen-associated molecular patterns, MAPs or PAMPs. For example, LPS, sensed in the extracellular space, is a MAMP that's characteristic of gram-negative bacteria, can be detected by TLR4. Or these molecular patterns can be host molecules that become altered. These are called danger-associated molecular patterns, or DAMPs. I suspect you've heard about some of these already, perhaps not characterized as such, but for example, in apoptosis, if cytochrome C comes out of the mitochondria, that is a trigger for apoptosis. That's a danger-associated molecular pattern because it is changing its compartmentalization from the mitochondria to the cytosol. It should not be there, and that can trigger apoptosis through APAF and caspase 9. Similarly, there are many DAMPs that are detected by inflammasomes that I'm going to talk about today. Inflammasomes actually tend to be a more complex manifestation of these categories, in that when they are detecting PAMPs or DAMPs, they're often detecting them as a marker of a virulence trait. So the immune system may be detecting LPS in the cytosol, but it's not actually less interested in the LPS than the LPS as a marker that the bacterium has invaded the cytosol. So there's like a level of interpretation that I think is greater than extracellular detection. So if TLR4 is detecting LPS in the extracellular space, that means there's a bacterium around. We don't know if it's dangerous. If there's LPS in the cytosol, that confers a different level of threat and danger posed by the microorganism. So I'll talk about all of the examples for inflammasomes. And I already said that. So there's many, and I'm going to be really focusing on pathogens rather than autoimmunity or immunopathology for inflammasome sensors. They're designed to detect bacteria, viruses, fungi, eukaryotes, and most of the studies have focused on these first two categories. And just overall pattern recognition receptors, here are the various categories of them. Inflammasomes fall in these last categories. Most are nod-like receptors, and then we have some examples of AIM2 and TRIM family inflammasomes. So here's another way to look at all those sensors diagrammed. We have a lot of toll-like receptors, C-type lectin receptors surveying the extracellular or vacuolar space. Inflammasomes are members of the cytosolic sensors. There's a lot of these cytosolic sensors that are poorly understood. We're going to be focusing on these ones, but just to put them in context of what they sense or how they act in comparison to other cytosolic sensors. Some cytosolic sensors like Rigi MDA5 cGas detect nucleic acids and trigger a transcriptional response, a type 1 interferon response that's very good against viral infection. Other cytosolic sensors like NOD1 and NOD2 detect bacterial peptidic glycan fragments and also trigger a transcriptional response or perhaps autophagy. Inflammasomes, the center category here, are different in that they signal to activate caspase-1, or the caspase-11 pathway is also included in this. And these cause post-translational activation primarily. So instead of causing new genes to be expressed, transcribed, and translated, these caspases are acting on already existing proteins, cleaving them. These are proteases, caspase 1 and 11, cleaving them to cause immunologic responses. So I also want to put these, some of the agonists, and I've just listed them all out here. I'll be going through them in more depth, but I want to point out that many of the agonists for the cytosolic sensors are actually the same molecules that are detected by the toll-like receptors that are looking on the extracellular or the vacuolar space. So often the same molecule is being detected in two different compartments with very different responses. So like I was mentioning, LPS, if detected by TLR4, will drive a transcriptional response. But if LPS is detected by caspase-11 in the cytosolic space, the cell explodes in the process of pyroptosis, which we'll cover more in a minute. So the interpretation of what LPS means and how should a cell respond to it really depends upon where it is in what compartment. And this is just summarizing what I said in a text format. I'm also happy to share these slides out with all of you. I also want to cover one of the important signaling domains that inflammasomes use to activate caspase-1, which are a subfamily of the death domain family. Death domains are small globular domains involved in signaling, typically in apoptosis or in inflammatory pathways. There are 4 different subtypes: death domains, death effector domains, caspase activation recruitment domains, and pyrin domains. And it's these latter 2 that are used by inflammasomes. And all of these 4 different subtypes typically undergo what's called homotypic interactions, so that a card of, for example, this NLR sensor will bind to the card of its downstream, for in this case, this diagram of a caspase. Or often what is seen is that there are adapter proteins that will allow, for example, a pyrin domain from an NLR inflammasome to bind to an adapter which amplifies the signal by polymerizing. In the inflammasome signaling pathway, this adapter is called ASC, which stands for apoptotic spec protein. It's called a spec protein because it polymerizes into a very large structure in the cell, this little 2-domain protein. And then the card domain of ASC can interact with the card domain of caspase 1. So you can get amplifying adapters converting one type of death domain family into another, bridging the interaction. But it's important to point out that not every card interacts with every card. So going back to this diagram of all of the pattern recognition receptors, if you look at all these pink circles in the diagrams, all of these are card domains, and all of these dark purple circles are pyrin domains. So for example, the card domains of RIG-I and MDA5 bind to the card domain of MAVs, but do not bind to the card domain of caspase 1. Card domain of NLRC4 binds to the card domain of caspase 1, but not to the card domain of MAVs. So cards bind to cards, but not all cards bind to all cards. There's specificity. So starting at the top with inflammasomes. Inflammasomes are defined as sensors or platforms that activate caspase 1. So sometimes this definition includes the ASC adapter, because that is part of the protein complex that is required to get to caspase 1. And sometimes the term is simply used for the signaling sensors in the NLRs. And it's a term that's used very variably in the literature. This is the official definition, but oftentimes people will include caspase 1 in the term inflammasome as well. So what are the, moving down the line to caspase 1, once caspase 1 is activated, this activates its protease domain. It's a specific protease that cleaves after specific aspartic residues only in very specific proteins. It was first identified as the protease that cleaved and activated IL-1 beta. It was first called interleukin converting enzyme, or ICE, because of this. Cleaving the first interleukin to its mature and active form. So pro-IL-1 beta and pro-IL-18 both cannot bind to their receptor. They are unique cytokines in that they do not have a secretion signal. So all other cytokines are translated into the endoplasmic reticulum and then secreted through the Golgi and exported from the cell. Pro-IL-1 and pro-IL-18 are cytosolic proteins. They don't have a dedicated secretion signal. They hang around the cytosol until caspase 1 cleaves them when it's activated, and then they form the mature receptor cytokines, IL-1 beta, but still they are inside the cell and they need to be released from that cell. And the way they are released is that caspase 1 activates a protein called gasdermin D, which forms a pore, which I'll have more diagrams of later. And that causes the cell to swell and lyse in a process we call pyroptosis. So pyroptosis is defined as the lytic cell death caused by a gasdermin. So the cytokines can actually be released through the gasdermin pore directly or through the membrane rupture that we were just talking about in the previous talk. So And actually, the gasdermin pore, I should have included a pore diagram here. Imagine a nice donut ring here, the gasdermin pore. It actually has biophysical properties that attract IL-1 and IL-18 by charge interactions to kind of electrically attract them to the pore and spit them out of the cell. So the pore itself efficiently releases these cytokines from the cell more efficiently than it releases other proteins. And the gasdermin pore is very large, allowing the passage of small proteins like IL-1 beta and IL-18. Caspase-1, so these are the three major substrates of caspase-1. Over the years, these three have held true no matter what. So these are real bona fide substrates. Over the years, there are many other proteins that have been proposed to also be cleaved by caspase-1. And many of them still await proving that they are physiologically relevant cleavage events. There's actually huge lists of hundreds of proteins in some proteomic papers that potentially can be cleaved by caspase-1. I'm going to mention a few of the targets here because they are probably bona fide targets. So one is that actually above the point of caspase-1 at the ASC polymer, ASC can recruit caspase-8 through non-homotypic death domain interactions, binding the DED domain of caspase-8 to, I believe, the pyrin domain of ASC, or maybe it's the card, I might remember wrong. Regardless, ASC can actually activate caspase-8. This probably only becomes relevant if a pathogen somehow inhibits caspase-1. So this pathway is slower than the caspase-1 to gasdermin D pyroptosis pathway. Caspase-1 can also cleave and activate BID, which then activates BAC and BACs, which are the mitochondrial pores. So many steps in this arrow that release cytochrome C, bind to APAF, which is related to the nod-like receptors, which then activates caspase-9. So this actually pretty quickly. This pathway is, again, probably slower than the gasdermin D pyroptosis pathway in most cells. Then caspase-8 or caspase-9 can activate caspase-3 and cause apoptosis. And this is probably a backup pathway. Like I mentioned, in case a pathogen was attacking and inhibiting caspase-1 or gasdermin D, then the cell could still achieve a different type of cell death to eliminate the cell if it's an infected cell. And these pathways in vitro are very solidly shown, but the physiologic relevance in vivo has some good evidence, but it's not as strong as the caspase-1 gasdermin D pathway. And these are really emerging pathways. Finally, caspase-1 can also cleave and activate caspase-7, which is another apoptotic executioner. Interestingly, it doesn't cleave caspase-3 directly. So caspase-1 is going to kind of work through these other upstream caspases, presumably because it's going to be integrating other cellular regulatory circumstances that may impinge on these for the cell to decide whether it's going to undergo apoptosis or not. All right, now I'm going to move on to inflammasomes and talk about each one and what they sense and what they do. So inflammasomes, as I mentioned, are the platforms for caspase-1 activation. They come in several different flavors. We have AIM2, we have several nod-like receptors, and I forgot to add here, pyrin, which is a TRIM family protein. Inflammasomes, as I mentioned, either card, which the card can directly activate caspase-1, or it can also interact with ASC and be amplified and activate caspase-1 indirectly. So two pathways for these card-containing inflammasomes, whereas pyrin-containing inflammasomes require the ASC adapter. If ASC is deleted, then these inflammasomes do not signal at all. Oh, and I want to go back to these backup pathways real quick. One other thing I wanted to say is, I mentioned the evolved function of these apoptotic backup pathways is probably to protect against pathogens that develop inhibitory virulence factors against caspase-1 and gastromin D. But actually in research, we typically see these pathways when we're studying knockout cells. So when someone makes a gastromin D knockout cell and then finds that the cells are still dying, but now they have apoptotic morphology. In my mind, as someone who thinks mostly about inflammasomes and pathogenesis, that sort of this knockout is simulating the effect that a cell would experience if it had a pathogen with a gastromin D inhibitory virulence factor. A different way to think about some of these backup pathways. Nod-like receptors oligomerize. This is a monomer of a nod-like receptor. When they're activated, they oligomerize into a hub. This can be 7 to 11 different NLRs, depending on the inflammasome. And what this oligomerization does is it clusters the pink card domains in the middle. These can then bind to either the ASC adapter or caspase-1, bringing caspase-1 in close proximity with other caspase molecules. These then dimerize and cleave each other between the P10 and P20 domains in the middle of the caspase, and this strongly activates the protease activity of the caspase. So this is another diagram that's just summarizing the inflammasomes I'm going to talk about in more depth today. AIM2, pyrin, NLRP3, NLRP1, and NLRC4 innate inflammasomes, as well as the caspase 11 side. So AIM2 first is perhaps the simplest inflammasome, because it has a HIN200 domain. These domains from other proteins are known to bind DNA, double-stranded DNA. That HIN200 domain is hooked to a pyrin domain. So it's a very small, simplistic inflammasome. When double-stranded DNA is present in the cytosol, the HIN200 domain binds to it, clusters on this DNA, that brings pyrin domains of neighboring inflammasomes in proximity to each other. Imagine another AIM2 kind of offset down here. These pyrin domains would be very close. And that can bind to ASC and initiate ASC polymerization. AIM2 is the founding member of actually a larger family of proteins that have this or similar domain architecture, where it's 1 or 2 pyrin domains and 1 or 2 HIN200 domains. Some of these family members actually are missing one or the other. They're all clustered in a genomic loci. So there's a family of these that seem to all bind DNA and have pyrin domains, but surprisingly, AIM2 seems to be the most important of them in detecting DNA in the cytosolic compartment. And AIM2 is good at detecting viruses whose genomes are double-stranded DNA, or bacteria that have lysed in the cytosol and released DNA. And this often happens if the bacterium is being attacked by what are called GBPs, guanylate binding proteins, which are essentially molecular cages almost that wrap around bacteria and somehow lyse bacteria in the cytosol. So Francisella is a bacterium that is attacked by these GBPs. This lyses the DNA comes out of the bacterium, and then AIM2 detects it. Here's a diagram of the AIM2 family from different species. What you can see is if you look closely. AIM2 is conserved in just about every organism, most organisms. It's the most conserved family member. And then the other family members are really pleiotropic between different species. So in humans, we have 4 of these AIM2-like receptors. Mice have 13 of them. And AIM2 is really the one that seems to be directly evolutionarily conserved. And what these other family members do is more contentious. There's a lot of literature on them. Some of them, IFI16 here is said to detect actually HIV infection. Perhaps the N200 domain is binding double-stranded RNA instead of double-stranded DNA. Or perhaps it's detecting something in the reverse transcriptase reaction. Some of these may be inhibitory molecules that are regulating AIM2, but really they're quite understudied in comparison to AIM2. Okay. Now I'm going to move on to the NLR inflammasomes. I'm going to start with NLRC4, perhaps because it's my favorite inflammasome, but also perhaps because we know the most about it and exactly what it does. So the NLRC4 inflammasome detects the activity of bacterial secretion systems, type 3 secretion systems, but also type 4 secretion systems. So type 3 and 4 secretion systems are virulence factors that bacteria use to intoxicate cells. So if this is a host cell, and this is a bacterium, the type 3 secretion system functions as a molecular syringe, which injects bacterial virulence, what are called effector proteins, from the bacterial cytosol through this hollow channel, needle complex is called, into the cytosol of host cells. And there, these proteins can interact with host proteins to reprogram something about cell physiology to benefit the pathogen. And bacteria can inject as few as 3 or 4 proteins, but as many as 20, 25 different proteins into the cytosol. And if you think about what a pathogen can do with 25 different virulence factors injected into the cytosol, it's pretty scary. Think about influenza that has I think it's like 10 or 12 proteins in total that makes up the virus. And here we have bacteria injecting 20 different proteins to reprogram host cells. So they can really commandeer and take over a cell and make it do essentially anything they want. And anything they want can be very different. So Salmonella uses its virulence factors to, for example, activate actin reorganization and cause invasion in epithelial cells, whereas Yersinia injects virulence factors to shut down actin and prevent neutrophils and macrophages from phagocytosing the bacteria. So you can use the same thing to be an extracellular pathogen as Yersinia or an intracellular pathogen like Salmonella. And then and about half of all gram-negative pathogens use one of these systems to reprogram our cells. So there's a lot of threat out there. The way the innate immune system detects these type 3 secretion systems is actually indirectly. So there are 3 different ways, all of them by detecting aberrant injection, translocation of proteins into the host cell cytosol. So we're actually not detecting the specific effector proteins that are being injected because they're variable between pathogens. We would have to have a specific sensor for every single pathogen. So instead, we have a single sensor that has the capacity to detect all type 3 secretion systems by 3 different routes. One of them is that takes advantage of the evolutionary relationship between the virulence type 3 secretion system and the flagellar type 3 secretion system. So these secretion systems are evolutionarily related. To make a flagellum, the bacteria exports flagellin monomers through this long hollow tube. They polymerize at the end of the flagellum and make this very long whip-like structure that then spins around. because this secretion signals and the biophysical properties of the flagellin protein have to allow it to be secreted and to transit through this long hollow tube, every once in a while, it can accidentally be recognized by the virulence type 3 secretion system and then injected into the host cell cytosol. So this is not what the bacteria want to do, but because evolution must combat biophysics essentially to prevent this from happening, it's very difficult for them to be perfect and not accidentally inject one flagellin molecule with every, say, 10,000 virulence factor proteins they inject. We actually don't know the ratio, but that is my guess. Something very rare. The other way that we detect this type 3 secretion system is by actually detecting two of the components of the apparatus itself. One of them is the rod protein. This protein, this is the assembly mechanism of how this needle complex is assembled. First, the bacteria makes this hollow shell, and then they secrete this rod protein into the middle, and it forms a spiral tube. So it has secretion signals on it. Then they secrete the needle that then polymerizes to form this short pilus. Both of these proteins are not supposed to enter the cytosol, but they can be accidentally, again, secreted and injected all the way across. This is not something the bacterium wants to do, but we think that biophysics prevents it from excluding these proteins completely. Now, once the rod, needle, or flagellin is injected, they can be bound by specific receptors called napes. There's one for the needle, one for the rod, and two for flagellin. These are the mouse genes. In humans, this seems to be condensed into a single protein that detects all three. These napes are actually NLRs. They look very much like NLRC4. Then they activate NLRC4, which then clusters and activates caspase-1. So we know a lot about the structure of the nape NLRC4 inflammasome. So this is what a nape looks like, an inactive nape. There's this, actually, I'll tell you what the domains are here. We have an NLRC4, there's a card domain. In the napes, there are 3 BIR domains, baculovirus inhibitor apoptosis repeat domains. They're obviously not inhibiting apoptosis in these proteins, but they were originally identified from a baculovirus. Anyway, they're small circular domains. Then there is the oligomerization domain shown in blue here, and then there's a leucine-rich repeat domain. As this folds, so the oligomerization domain is actually composed of several subdomains shown here in orange, blue, yellow, and green. And then the leucine-rich repeat kind of actually wraps around and folds over and grabs on and holds this oligomerization domain in the inactive state. When flagellin binds to this, to the nape, what happens is there's a conformational change such that the leucine-rich repeat, there's a hinge right here, and the leucine-rich repeat kind of cranks to the side like that. And you can see, actually, if you watch this silly cartoon I've made and just watch these helices, they don't move, but the leucine-rich repeat domain is cranking open. What that does is it exposes this part of the oligomerization domain, which is normally hidden in the closed structure. Flagellin binds here in purple, and it actually looks like it's kind of like a pin stuck in the jaw of this nape, preventing it from cranking back into the closed position. It's like holding it open. So once this nape is activated, the polymerization interface is exposed. What this does is it binds to an NLRC4 monomer. So NLRC4, imagine it existing in this phase. This back interface is this side of NLRC4, so it can dock on the nape, this receptor receiver domain after the oligomerization interface is exposed, it can bind. That binding actually triggers the same conformational change. So this teal leucine-rich repeat domain used to be bound over here. When it binds to the nape, it cranks open, exposing the next oligomerization interface right here. That can bind to another NLRC4, which docks, cranks open, exposes another interface, docks, cranks open. And this just goes on around the circle. The amazing thing about this structure from Hao Wu and Russell Vance and Gigi Chai's labs is that a single flagellin monomer causes the oligomerization of 10 NLRC4, which then expose their card domains. This will then cause both ASC polymerization and caspase-1 polymerization, caspase-1 activation. Caspase-1 is a protease that can cleave many of its target proteins. And so there's this massive amplification built into the system, such allowing one flagellin monomer to essentially massively amplify into through 2 polymerization steps and a catalytic protease step to then cleave a whole bunch of IL-1 beta, a whole bunch of IL-18, and a lot of gas German D, which will then poke holes in the cell. So the sensitivity of this system is really potentially.
We lose our sound.
Hello? Did we lose me? Can you hear me? Am I not?
No, now it's okay.
I'm back. I'm back. Okay.
Yes, you are back.
My voice is being transmitted from across continents. It's really amazing that we can do this, isn't it? And just one little glitch. Okay. So potential sensitivity of one protein molecule. And I also want to draw your attention. One of the reasons I'm going through this in depth is because it's actually one of the simpler inflammasomes, and some of the concepts in here are going to translate to NLRP3, which we're about to move on to. So one of the things I want to draw your attention to is that they're actually in this inflammasome oligomer, there are 2 interfaces, 2 polymerization interfaces. One here, which is the one that the next NLRC4 docks to, but then once it cranks open, the back of the leucine-rich repeat interacts with the front of the leucine-rich repeat from the other molecule, which holds the structure open and provides a double interface to make a stronger structure. And we're going to see that play out differently in NLRP3. So let us move on to another inflammasome, NLRP3. Sorry, I want to just say one more thing about NLRC4. The importance of actually completing this pathway is that if a cell is injecting virulence factors to reprogram, say, a macrophage, that macrophage is now an agent of the enemy, and it should, the best thing we can do is destroy that cell, because it's probably infected with bacteria and serving as a replicative niche where bacteria are multiplying. So you need to destroy the cell via pyroptosis. And you also need to alert the rest of the immune system that we have an intracellular pathogen that's very dangerous. Or we have a type III secretion system using pathogen that's reprogramming our cells. So once it's essentially, the cell assumes that it has lost the fight once it's been injected, and it's going to explode to get rid of itself so that other immune cells can take up the fight in a more effective manner. Okay, moving on to NLRP3. NLRP3 is probably the most important inflammasome. It is one of the most, I think it's probably the most complex inflammasome. It is the most studied inflammasome, and it is the one, actually, I think, one of the ones that we know the most about and the least about at the same time. So NLRP3, it requires licensing and priming, which I'll describe in more detail. It responds to many different perturbations. I like to say that it responds to half of the sigma catalog, but a very specific half. So it'll respond to toxins, crystals, ATP are some of the primary agonists that are used. The toxins like the nigericin or pore-forming toxins release potassium from the cell. And at one point, my bet was on potassium. This is an old slide. At one point, my bet was on potassium as being the terminal event that was sensed by NLRP3. Now I've just kind of given up on guessing, honestly. At one point, lysosomal disruption was thought to be one of the signals. This has gone out of favor, I think, in the field. But if you read papers, you'll see things about cathepsin's release into the cytosol as potentially being an agonist. Certainly, the toxins, when nigericin or pore-forming toxins do cause potassium efflux, if you block that, then NLRP3 is not activated in response to those same toxins. So this is part of the pathway, at least for the toxins. Other hypotheses, and I think this might come a lot from the crystals like alum or monosodium urate, is that mitochondrial dysfunction and mitochondrial reactive oxygen generation can cause NLRP3 to activate. This is all extremely controversial. Exactly what the terminal signal is unknown. I have often thought about NLRP3 as a sensor of cellular catastrophe. So one of the things that these all have in common is if a cell is perturbed enough that it cannot maintain its sodium-potassium gradient across the membrane, it's really like a terminal catastrophic event in cell biology. The cell is extremely messed up. If the cell is perturbed enough so that mitochondria are spewing ROS, reactive oxygen species, into the cytosol, that could also be a sign that the cell has really reached a terminal catastrophic event. So NLRP3 could be a sensor of catastrophe. Or it could be that these catastrophic events are mimicking something else that is the real signal for NLRP3. So because it seems to be activated in a kind of non-specific way, many different pathways, and also activated in the context of catastrophic cellular events, there are many, many ways to get to that point. So many different pathogens will cause NLRP3 to activate, but also many different immunopathologic conditions will cause NLRP3 to activate. And if you're thinking about like inflammatory bowel disease or sepsis or something like that, you can imagine NLRP3 activating just due to the overall inflammatory environment and basic tissue homeostasis becoming very altered in a destructive way. NLRP3 may activate inappropriately, release a bunch of inflammatory IL-1 beta, and then cause more inflammation. One of the primary agonists here, alum, or sorry, not alum, monosodium urate, is urate crystals, and those form when patients have gout. So crystals form in their joints, and NLRP3 is thought to be the pathologic driver of gout inflammation, causing IL-1 beta release, and IL-1 beta then causes joint inflammation and gout. And antagonists of IL-1 beta have been useful in treating patients with gout. Another common agonist that you'll see used is ATP. This binds to the purinergic receptor P2X7, which opens a potassium. Sometimes it's said to be a sodium channel, sometimes a potassium channel, and I I'm forgetting which it was actually right now. I think it's mostly said to be a potassium channel, but I think I read once that it was a sodium channel. Anyway, it opens a channel that breaks this membrane ion gradient. And it requires extremely high concentrations of ATP. The type that perhaps if a macrophage was sitting next to an intact cell and that cell lysed, huge amounts of ATP might be experienced by the macrophage, and that could be considered a danger signal. Low amounts of ATP bind to other purinergic receptors and are important for apoptosis. They are the find me signals that macrophages use to follow and find an apoptotic body. So the amount of regulation that the immune system has put into NLRP3 is really immense. This is a figure. I'm not going to go through all this, but I'm putting this up just to illustrate. This is a figure from an old review from ours that has like bazillions of things that can impinge upon the NLRP3 inflammasome. Probably half of this is actually not quite right. It might be artifacts of non-specific inhibitors or experimental systems, but you can see that it is very, very complicated. I'm going to talk about what I think are the most solid recent advances in the NLRP3 field, and that is that NLRP3 requires NEC7 licensing, which is so there are multiple steps in NLRP3 activation. First, resting NLRP3 protein is resistant to being activated. So if you put any one of those agonists that I mentioned onto a cell, it can cause that catastrophic potassium loss, for example, but NLRP3 will not activate unless it is first primed. What is priming? We don't know. We know how to prime, and we have many hypotheses as to what priming is. How to prime NLRP3, it is accomplished by any TLR, essentially, or TNF, any way to activate NF-kappa B signaling. Priming is both post-translational, and this may be accomplished by adding a phosphate, by phosphorylating NLRP3, or removing a ubiquitin from NLRP3. So something may ubiquitinate it and hold it in the inactive state, remove the ubiquitin, and it's now primed and ready to be activated, or adding the phosphate, or both. One of the things about the NLRP3 literature is there's just a lot of information and claims, and it's hard to integrate them all into one cohesive whole. So once primed, the other aspect of priming is that NF-kappa B activation will induce a transcriptional induction of NLRP3 as well, which increases its sensitivity. But you definitely need some sort of post-translational event, either phosphorylation or deubiquitination. Then if NLRP3 is primed, it can be activated by one of those agonists through unknown molecular mechanisms, but the activation should induce the same rotation that I showed you in the nape. So if you imagine a hinge right here, where I'm pointing, then this nucleotide binding domain hinges up, exposing these two parts of the oligomerization domain, which are the oligomerization interface. Then these can bind to another NLRP3. However, remember I said for NLRC4, the leucine-rich repeat bound to the backside of the next leucine-rich repeat. In NLRP3, it's actually shorter, and it's too short to reach. So there is no binding interface here in unlicensed NLRP3. So without this additional protein called NEC7, nothing happens. It does not activate. However, if NEC7 is present, I'm going to tell you what NEC7 is in a second, bear with me. If NEC7 is present and available, then NLRP3 is licensed. And what that is that NEC7 binds right here in the midst of the leucine-rich repeat horseshoe. And it can either, I don't know if it's binding to the unprimed and then it gets primed, or if you can get primed and then licensed, I suspect that it can happen in either direction as drawn here. But then once NLRP3 is primed and licensed, if then it is activated, and you get that rotation, now the next NLRP3 can bind, crank open, oligomerize, and connect with the previous one at 2 interfaces, and then proceed around the hub to form the inflammasome. So 1 interface, 2 interfaces. One of the things about the NEX7 or the NLRP3 literature, as I mentioned, is there is just a lot of many publications, and the reason why I'm highlighting NEX7 is because this is one of the regulatory steps that is advanced to having structural data. So we have how we published the structure of NLRP3 binding NEX7, showing that it can bridge this second interface, which has made me believe that this is really important for NLRP3, and that's why I'm highlighting it. What is NEX7 and why is NEX7 used to license NLRP3? It's a 2-domain protein. It's a kinase. So it's only the C lobe that is bound to NLRP3. This N lobe is hanging off, presumably, to the side. So NEX7 is a NEMO-activated kinase. It's part of the mitosis process, actually. So it's a kinase that is activated during mitosis to organize the mitotic spindle. So during mitosis, it localizes to the centrosome, organizes the mitotic spindle, and is participating in that process. And probably during mitosis, NEX7, because it is in the centrosome and being sequestered and used, is not available for NLRP3. Therefore, what using NEX7 licensing does is it permits NLRP3 activation during interphase. So during interphase, NEX7 is free in the cytosol. During cell growth, DNA synthesis, cell growth again, NEX7 is free and NLRP3 can find it. But during mitosis, NEX7 is essentially sequestered at the centrosome. NLRP3 cannot find it. Therefore, if the activation signals came through for NLRP3, it would not be able to complete the oligomerization into the hub. Why, then, is this important? Why is a inflammasome that's very important for presumably sensing pathogens be regulated during mitosis? We don't know why, but the speculation is that, or my speculation, I think this is the general speculation, is that something is happening in mitosis that causes a false activation event of NLRP3, or causes it likely to be falsely activated. If we can ever figure out what the actual molecular activating signals are for NLRP3, I think this is going to make a lot of sense. And we'll say, oh my goodness, of course, during mitosis, blah, 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 whatever those signals are going to be present inappropriately and not as a marker of infection, because mitosis is an extremely dramatic event in the cell. Actually, if you think about catastrophic events in the cell, mitosis is like dramatic in the extreme. Cells organizing all their chromosomes and then pulling them to another part of the cell, splitting the cell in half. That is really dramatic. So something about the dramatic events in mitosis, I think, causes a false positive for NLRP3. Thus, it's useful to have this revocable licensing where NLRP3 can be in the ready to go state, or just shut off, license revoke, and it can go back and forth easily. Okay, I'm going to move on to NLRP1 next. Our understanding of NLRP1 has recently come into fruition from Russell Vance's lab and Machovshin's lab. So NLRP1 is an interesting modification of the typical NLR domain structure. So normally NLRs have a signaling domain, a oligomerization domain, and a leucine repeat. P1 has an additional domain here called the find domain, and then an additional signaling domain at the end, a card domain. So in humans, we've got a pyrin and a card. In mice, actually, that pyrin is missing, and we just have the card at the end. And it turns out that it's the card at the opposite end of normal that is the signaling domain. So this pyrin domain is actually dispensable. It's the card domain that is the signaling domain. And in mice, same. Humans have one copy of NLRP1. Mice, black 6 mice, have 2 functional copies and one that's probably a pseudogene, but it's actually quite variable in mice. There's, I think, up to 5 copies of NLRP1 in different inbred mice strains, and as few as 1 in others. We don't know, I think, the physiologic activator of NLRP1. There are actually, no, I take that back. There are proteases that are said to activate NLRP1 in humans from viruses. And in mice, it's best understood, the anthrax lethal toxin is one of the best characterized agonists that activates NLRP1. And I will go into that in depth in the next slide. Toxoplasma can also activate NLRP1 via less clear means. And I also want to mention the anthrax lethal toxin activates NLRP1, but only the NLRP1 allele, only the NLRP1B allele, and from certain mouse strains. So it does not work in black 6 mice, the immunologist's favorite mouse, but it works in 129 mice, the genetics, the historically best genetic mouse. Okay, coming back to this find domain, what this find domain, it was named function to find. This II was like a Roman numeral 2, function to find domain. So the function to find domain, the function is now known. It is an autoprotease domain, which cleaves itself to separate the peptide bond. However, the cleaved portion remains associated with the rest of the NLR. And the reason for that I will explain in a second. So anthrax toxin, coming back to the agonist, anthrax lethal toxin is a toxin that anthrax bacterium secretes. It is also a protease. And actually there is a surprising number of proteases in this pathway. We have one protease step here in autoproteolysis NLRP1. Another, the agonist, anthrax lethal toxin, is a protease. Anthrax injects this into cells in order to cleave and inactivate MAP kinases to shut off transcriptional signaling. So anthrax wants to shut down the MAP kinases. We would like to detect when our MAP kinases are being attacked, we mammals, mice in this case, we mice. And NLRP1 accomplishes this. So this is another diagram of the closed form of NLRP1, the blue oligomerization domain, the green leucine repeat, coming into these 2 orange domains, which I'm coloring different colors here because they're cleaved, but as drawn here, the small domain remains associated. And the whole domain is called the find, and then the 2 individual domains are UPA and, oh no, I'm forgetting the other acronym. Anyway, the important one here is the UPA that is attached to the card domain. So, and I'm marking the protease with a little star here. So there's a cleavage event in the resting NLRP1. And what NLRP1 has is essentially it has a lure domain in its amino terminus, which is a decoy target that contains sequences that lethal toxin is looking for to cleave a protein. So lethal toxin cleaves NLRP1, in its little amino terminal Lure domain. Then basic cell biology takes a role. And in basic cell biology, we have ubiquitinases in the cell that are looking for broken proteins. Every protein should have a methionine at its amino terminus. That's how all proteins are made. So if there is a protein that has an amino terminus that is not a methionine, that means that something unusual is happening. And in most cases, it means the protein is old, broken, it got cleaved inappropriately or by some protease something. And it's time for that protein to be recycled. So old broken proteases, or old broken proteins, are identified by what are called N-end rule ubiquitinases. The N terminus, the N-end, N-terminal rule is that it should be a methionine. And if it's not, we're going to ubiquitinate it, polyubiquitinate it, and send it to the proteasome to be recycled. So this is exactly what happens after anthrax lethal toxin cleaves NLRP1. The whole thing is sent for destruction. Protease number 3, the proteasome, chews up NLRP1 down to amino acids or peptides, I forget. And what this happens is that if the proteasome essentially unfolds the protein, pulling it through one amino acid at a time, but when it gets to the UPA domain, there's a break in the peptide bond. So as it takes apart the rest of this protein, the UPA just falls off and is released. It is now liberated and free in the cytosol. So this liberated UPA card is actually then oligomerizes, the UPA domain oligomerizes and forms an inflammasome. So strangely, the inflammasome complex here is not the oligomerized NLR. It is this little piece at the end that falls off as the NLR is being destroyed because something attacked it. So this inflammasome oligomerizes, presents clustered card domains that activate caspase-1. So the importance of this autoproteolysis is if it essentially enables this detection system. If this was intact, the whole UPA card would just be shuffled right into the proteasome and destroyed because there is this break in the peptide bond. Because the rest of the NLR holds the card, the UPA card, inactive and prevents it from oligomerizing. Once it's separated, the UPA card can now oligomerize and signal and form the inflammasome. So this strategy actually creates a little bit of a conundrum. I mentioned the N-end rule deals with old broken proteins. What happens when NLRP1 gets old? It'll go to the proteasome and liberate UPA cards. So hopefully only one happens at a time. If there's only one UPA card, it's not going to signal. It needs to cluster with many of them. But there is another regulatory step that is involved in this. We don't really understand what is going on actually at this step. But in a resting cell, NLRP1 is inhibited by another protease called DPP9, DPP8 or DPP9. They're similar proteases. These are dipeptyl proline proteases. Essentially, this protein is looking for proteins that have a proline at the second position from their amino terminus, and it cleaves after that proline. So it's another protease. Strangely, it seems like that protease activity is not required for activation step. Instead, the DPP9 holds this find domain, the two pieces of it, the UPA and the, I wish I could remember that, the other piece of the find domain. And then it actually binds to a third upacard that has been liberated. So if we have one liberated, it will dock here as well. And we do not know really what the function of this structure is, but we know that it's important, because if you delete DPP9, NLRP1 will spontaneously activate. If you inhibit DPP9 with small molecules called valboropro, which inhibit the protease activity, which oddly doesn't seem to be important, but perhaps displaces the structure when you add the inhibitor, NLRP1 will spontaneously activate. So there is some regulatory step here where if you get just a little bit of upacard, it'll come over here, bind to this ternary structure, be inhibited. Perhaps this whole thing is destroyed somehow by maybe autophagy to package it away from the cytosol. Maybe it's sent to the proteasome in some kind of clever manner to not release these. We don't know what's happening yet, but we do know that it's important. And again, once you get structural information like this, that shows you how the NLRP1 is docking in a very strange way to this protein, you know it's going to be real and important, but we don't know exactly why yet. Well, we do know why. We know, I think, why is to deal with the occasional recycled protein, but exactly how this all works is yet to be figured out. And so that's 4 different protease steps. Just want to highlight them again. The find cleavage, the agonist cleaving, the proteasome cleaving, and then this other protease, which doesn't seem to be important for its protease activity. So a lot of protease sensing in the NLRP1 pathway. So, it's best shown for lethal toxin, but as I mentioned, there are viral proteases that are now also detected by NLRP1, the human gene. There are also other ways to cause proteasomal degradation. There's a bacterial type III secretion effector that is said to ubiquitinate NLRP1, probably in the same decoy targeting strategy. So there's a Shigella effector that ubiquitinates NLRP1 and sends it to the proteasome, releasing the upacard. So it doesn't have to be the end rule. Essentially, any way this thing can be degraded. Anyway, it can be sent to the proteasome, or if it was just, if a protease, if a pathogen actually had a protease that attacked it in a way that really unraveled the whole protein, that would also release the upacard. Okay, that's NLRP1. Move on to pyrin next. Pyrin is a member of the TRIM family. There are 80 different or so tripartite motif family members. They do many different things. Many of them are involved in interferon regulation. Pyrin is the only one of this family that has an inflammasome signaling domain. None of them have. So it's really unique amongst the TRIM in that it is an inflammasome. And what pyrin does is it's a guard, essentially. When I say guard, is meaning that it monitors for dysregulation of Rho GTPases. And it's the Rho GTPases that are regulating actin cytoskeletal remodeling, so Rho A. Many Rho A is often targeted by toxins or other virulence factors for inactivation. Pathogens like to inactivate Rho A because it is required for phagocytosis. So for example, Yersinia is a bacteria, as I mentioned, that injects type III secretion virulence factors. It wants to stay outside of the cell. So it's injecting neutrophils with anti-actin virulence factors, such as YOP-T, which will attack the Rho-RAC GTPases, and YOP-E, which will also attack the same Rho-RAC GTPases. So it's double attacking our actin regulatory GTPases, preventing the phagocytic cup from extending around the bacteria, preventing phagocytosis, which allows the bacteria to be an extracellular pathogen. This is really disastrous for the immune system. If your neutrophils cannot eat bacteria, you're essentially neutropenic, and that's like the worst thing that can happen during a bacterial infection, which is probably why Yersinia is the causative agent of the plague and has from time to time ravaged huge fractions of the world's population. It's one of the most virulent bacteria on the planet. Because it inhibits the Rho GTPases, but it also inhibits pyrin from detecting that has occurred. So what happens is, here's a diagram of pyrin. Human and mouse pyrin are slightly different from each other, but they're both sensing this Rho perturbations. So normally, Rho GTPases, when activated, should polymerize actin and also activate downstream kinases such as protein kinase N, PKN, which then phosphorylates other proteins that are involved in the processes. Yersinia, when this successfully occurs, protein kinase N, PKN, also phosphorylates pyrin, which holds it in an inactive form. So once it's phosphorylated, pyrin does not activate. What this is doing is PKN is telling pyrin that the Rho GTPase cascade has successfully signaled. However, Yersinia injects virulence factors which shut off these Rho GTPases. Therefore, you don't get PKN activation. Now pyrin sees that it has not been phosphorylated. Then it activates, exposing its pyrin domains to activate caspase 1. And pyrin exists as a dimer. They're just shown in slightly different shades here. But Yersinia, being the most deadly pathogen in the world, has evolved to prevent pyrin from detecting it with a third type 3 secretion effector called YAP-M. What YAP-M does is it grabs protein kinase N and then grabs pyrin and directs PKN to phosphorylate pyrin. So what Yersinia effectively does is prevents actin polymerization and simultaneously inactivates the sensor of actin polymerization. What the field is still looking for actually is the pathogen that prevents actin polymerization but doesn't inhibit. And this might be in the Burkholderia Cenocepacia complex, where there are actin inhibitory toxins that the bacteria release to inhibit actin polymerization. Next, we're going to move on to caspase 11, which is a parallel pathway to get to pyroptosis. So caspase 11 has a card domain and a caspase domain, just like caspase 1. And it detects LPS that is present in the cytosol. So when bacteria rupture and enter the site, because they're cytosol invasive, their LPS can be detected by caspase 11. This requires some effort because LPS is embedded in the bacterial surface. And actually, again, the GBPs that I mentioned earlier that attack bacterial surfaces appear to be important in revealing this LPS to caspase 11 for detection. So unlike caspase 1, which has these upstream sensors, caspase 11 does not have an upstream sensor. The LPS actually binds directly to the card domain. And actually, it binds to it on the bacterial surface. So caspase 11, once that bacterial surface has been disrupted by GBPs, it binds to the surface of bacteria and will coat the bacterium and activate the protease domain there. Caspase 11 can cleave gastromin D to cause pyroptosis, but unlike caspase 1, it seems to not be able to cleave the two cytokines. Why that is still a little bit of a mystery. However, the processes of pyroptosis, once you open a gasdermin pore, that starts letting potassium leak out of the cell, And it's just like if you treated the cell with a pore-forming toxin, this is going to progress towards catastrophe. And this can actually activate, NLRP3. The Gas-German D-pore will activate NLRP3, which is sensing that catastrophic loss of potassium. Then NLRP3 will activate caspase-1, which will activate the cytokines. Whether this pathway is physiologically relevant remains unknown. It's definitely detectable in vitro very easily, but whether it's important in vivo remains to be seen. Because this is a sort of strange way to activate caspase-1, this pathway has been called the non-canonical inflammasome pathway. Non-canonical meaning not normal or not the one we know about. So these, the inflammasomes that I've been talking about so far can be called canonical inflammasomes that make platforms that activate caspase-1. And caspase-11 can be called the non-canonical inflammasome to caspase-1. But then the term has also just simply been used to refer to the simple pathway of LPS detection. Caspase German D and pyroptosis has been called the non-canonical inflammasome pathway. It's a term that's used very loosely and non-precisely by different labs. The term non-canonical also just means not normal. So any way to activate caspase-1 could be called non-canonical. So other investigators have used that to refer to other ways to get caspase-1 to activate, not caspase-11. So it's a term that's a little bit confusingly used. In humans, in mice, we have one caspase-11. It's encoded directly adjacently to caspase-1. In humans, like Yusuf Ashwi here, my former postdoc, who's now got his own lab studying, he studies the caspase-11 pathway. We have caspase-4 and caspase-5, which are duplicate genes of caspase-11. All of these detect us. Why humans have two copies of the gene, we do not know. They seem to have different expression patterns in different cells, so there may be different regulatory aspects hooked to them. But one of the, I want to get on I'm going to get on my soapbox here for a minute and talk about one of the phenotypes that's interesting in the caspase-11 field, and it kind of illustrates pathogens evading these sensors and how pathogens that are evading sensors can give you intermediate phenotypes, whereas pathogens that don't evade can give you stronger phenotypes. So when we were studying caspase-11, we examined Burkholderia pseudomallei, which is a cytosol invasive bacterium. It's a highly virulent and potential biological weapon. And we did an experiment where we found that wild type mice would survive, whereas the caspase mice would succumb. At the same time, Fabio Rey's lab was doing the same experiments, and I'm going to regraph his 3 experiments because he did a more thorough job than we did. He used 3 different doses. And when he looked at a very low dose, he got the same response as we did. 25 bacteria would kill a caspase-11 knockout, but not a wild type mouse. These are 111s actually. But when he raised the dose to 100, now about half of the wild type mice died. And when he raised it to 200, all the wild type mice died. So you can see that caspase-1 and 11 here are giving you about 200 divided by 25, about an 8-fold change in the apparent lethality, which you would only detect if you used multiple doses. If you did the lazy experiment like we did and just use 1 dose, it looks like you have a really strong phenotype. But if you're thorough like Fabio, you realize that you have an 8-fold change in the lethal dose. And if you're inhaling Burkholderia pseudomallei, what are the chances that you're going to inhale? 25 versus 200. Who knows? But this is why Burkholderia pseudomallei is an incredibly virulent bug. When we were looking at a different bacterium, a relative called Burkholderia thailandensis, this bug cannot kill wild type mice at all. You can inject 2 times 10 to the 7th into the peritoneal cavity, and the mice will clear them in 1 day and survive. The caspase-11 mice die, and this graph kind of looks like this graph. But when we did lower doses, you can go down to 10 to the 4th, 10 to the 2nd. Actually, you can even just inject 1 bacterium into a caspase-11 knockout mouse, and it'll replicate out of control and kill that mouse in about a week. And what we think is that Burkholderia thailandensis has no ability to fight back against caspase-11. So it's totally wiped out from wild type mouse. But if you give it the ability to fight back against caspase-11 by deleting the caspase, it can just go wild and turn itself into a biological weapon like Burkholderia pseudomallei. Now we don't know how pseudomallei is evading caspase-11. I'm assuming that it is because it's so virulent, but we do know how Shigella is evading. So the 2 Burkholderia I was just talking about actually encode a similar type III secretion system to Shigella that allows them to invade the cytosol. So they're kind of a family. Shigella invades the cytosol, and one of the type III secretion effectors that it injects is called OSP-C3. And this was recently shown by Feng Xiao's lab. The mechanism of its inhibition of caspase-11 was recently shown by Feng Xiao's lab. It was originally shown that it inhibited by Sasakawa. So OSP-C3 binds to caspase-11 or caspase-4 in humans, and ADP boxinates it, which is a chemical reaction. It adds this ADP ribose to the caspase, which prevents it from interacting with gasdermin D and cleaving that protein to its active form. So Shigella really efficiently shuts off the sensors that are supposed to be detecting its LPS. It's also using other OSPs to shut off other caspases. And this is probably why Shigella is one of the most virulent diarrheal pathogens known to people. If you ingest 10 Shigella, you are going to get bloody diarrhea and be very, very unhappy. All right. I think I'm actually doing pretty good on my time here. I'm going to move now down to the targets of caspase 1 and 11, and I'm going to give really short overview of IL-1 and IL-18, and then I'm going to talk about the gasdermins at more length because they're more, there's a lot more being discovered about them right now. So beta is one of the first cytokines ever discovered. It causes fever. I think it was discovered because it causes fever. You can inject it into animals and they'll get a fever and inflammation. One of the things I like to do when I'm thinking about cytokines is just ask where the receptors are expressed. So if you look at the IL-1 receptor in bio-GPS, you can see it's expressed all over the place. Many different tissues express it, many different cell types. And one of its cardinal functions that is that it will cause local inflammation by interacting with endothelial cells, causing them to present their selectins, which allows leukocytes in the blood to adhere, to roll, and then they can follow chemokine signals into the tissue. It also causes vascular permeability. But IL-1 beta is typically referred to as a pleiotropic cytokine because it has many, many, many functions. This is just one of the most, probably one of the most prominent is local inflammation. IL-18, if you do that same bio-GPS, you see that, wow, NK cells and T cells are really the cells, and actually grossly in tissues, it's barely expressed. So IL-18 is a very specific cytokine, talking specifically to T cells, particularly Th1 cells, cytotoxic T cells, and innate lymphocytes, ILC1s, and natural killer cells. It induces them to produce interferon gamma, and IL-18 was actually first identified as interferon gamma inducing factor. Interferon gamma is very good at combating intracellular infections like listeria infections. And actually essentially every intracellular infection, interferon gamma is good against it. So IL-18, very good at driving interferon gamma. And it also has many other functions, but I think that's the number one. So next, I'm going to move on to pyroptosis and Cas-Dermans, talk more about Cas-Dermin D. So either caspase-1 or caspase-11 can cleave Cas-Dermin D, which is a two-domain protein. It has a pore-forming domain, a linker, and a regulatory domain, a repressor domain. So the repressor domain normally holds tight to the pore-forming domain and it keeps it inactive. However, once caspase-1 or 11 cleave that linker at that aspartate, the pore-forming domain is liberated. It oligomerizes. Actually, I've shown it incorrectly here. It oligomerizes in the cytosol, then binds to the membrane, undergoes a conformational change, and inserts into the membrane. So this wheel structure forms and then docks onto the membrane and then inserts. This creates a gigantic 10 to 15 or larger nanometer pore, which is large to allow small proteins like IL-1 beta to be released, which is only 4.5 nanometers in diameter. And as I mentioned before, there are charge properties of the inside of this pore that actually attract IL-1 beta to be spit out. At the same time, all small molecules from the cell are being released through this Cas-Dermin pore. Sodium, potassium, ATP, glutathione, everything is going to be released, and all small proteins. Just IL-1 might be coming out faster. Simultaneously, extracellular molecules are entering the cell down their electrochemical gradient. So sodium is rushing into the cell and causing swelling. This is just a diagram, or not diagram. This is a cryo-EM structure from Hao Wu's lab of one of the other Cas-Dermans, actually, but now we have a Cas-Dermin D structure that looks just the same. And you can see this really huge hole. Starts 18 nanometers. This was an older number. This causes swelling. Actually, now we know that this something between the pore, we used to think, sorry, I should go back here. We used to think that the swelling of the cell caused the membrane rupture. But as Peter was talking about just before I started my talk, Vishva Dixit's group recently discovered that another protein is in between the Cas-Dermin pore and the membrane rupture. And this protein is called NINJ1. Something, we do not know what, causes this very small protein after the Cas-Dermin D pore opens. Something causes this very small protein to oligomerize. It's a 2-transmembrane protein with an extracellular domain. Actually, I've flipped upside down here. There's an extracellular domain, a small cytosolic domain, and something causes this to oligomerize and disrupt membranes, and then that membrane rupture occurs. Without this, the cell will swell, but the membrane actually never ruptures. So anything bigger than the gas will not be released from the cell. So lysis requires NINJ1. There is a recent paper on bioarchives actually that says that NINJ1 is might be the glycine sensor. So it's been long known that if you put a very high concentration of glycine onto a macrophage and then activate caspase-1, that the pore will open, caspase will activate, but the cell will not rupture. And actually, this bioarchive paper is saying that without NINJ1, glycine is not sensed. So one of the things that I'm wondering is if this little cytosolic domain is actually a glycine sensor, sensing when the concentration of glycine in the cell, which should be high. If glycine is released from the cell, that could be a marker of irreversible membrane collapse or membrane damage through pores, and that might cause NINJ1 to oligomerize to take apart the cell further. Total speculation on my part there. So here, I just want to show you a nice little video showing pyroptosis. These are macrophages, and there's a red dye in the media that cannot enter when the membrane, you can see that red dye dramatically enters the cell and stains the cell red. So that's pyroptotic cells have this characteristic like large balloon like blown out structure morphology. So to put pyroptosis in the context of other types of cell death, necrosis is lytic cell death that is non-programmed. So physical, thermal, or chemical damage will cause a cell to lyse. It is not programmed by any of our proteins. Pyroptosis is programmed lytic cell death. As I've been mentioning, inflammasomes activate caspase 1 or caspase 11, 4 or 5 can activate to LPS, opening the gastrointestinal deep pore and causing lysis. Similarly, necrotosis is also programmed, but it's got a different signaling pathway, which you're hearing about in other lectures. So it's programmed, but it's lytic. Apoptosis, in contrast, is programmed. And then we also have netosis, neutrophil extracellular trap-related cell death, which is specific to neutrophils and a small fraction of macrophages, which is programmed and can be lytic or non-lytic. Okay, I'm going to now move on to a cell type specific manifestation of this pyroptotic cell death, or rather gastrointestinal D-induced and caspase 1 induced cell death that's been described recently, first by Wolf Teacher-Cart and Russell Vance's lab in intestinal epithelial cells. So intestinal epithelial cells line your gut, small, large intestine. They are a single columnar epithelial layer. This is a picture of an organoid, which is in culture, but if you in culture, intestinal epithelial cells can be induced to make spherical little balls where the inside of that sphere is the equivalent to the lumen of the gut, or you can focus single focal plane and it just looks like you're looking at a cross section of a gut with intestinal epithelial surrounding it. So when caspase 1 activates in intestinal epithelial cells, what happens is these cells will jump out of the monolayer into the lumen of the gut and remove themselves essentially from the body by diving into the poop stream to eliminate any infected pathogens. And this process is called extrusion. And here, Russell Vance has a really nice video. You can see a cell here. Watch this cell. Bloop. See how it just, this cell, they pop out of the monolayer into the lumen. Now, this organoid was treated with Flawtox. So every single cell in this organoid is being activated for caspase 1 at the same time. This just happens to be the first one that got a dose, and you can see really nicely how they extrude into the lumen. In a real infection, you would get just one cell at a time extruding, and only the cell that was infected by a bacteria. This extrusion after inflammasome activation happens by multiple pathways. Probably gastrointestinal D pore itself activates the extrusion process, but also Vance's lab showed that the ASC caspase 8 pathway can also, that backup pathway that I mentioned, can also cause it. And I suspect that the caspase 1 to BID pathway will also cause this extrusion to occur. Perhaps these occur on different timescales. So IEC extrusion is it's actually a very complex process. The cell the cell that is extruding must actively participate in its extrusion. It needs to reorganize its actin cytoskeleton and contract an actin-myosin ring to squeeze itself out of the monolayer. Meanwhile, its neighboring cells are also squeezing to push it, but the cell that is extruding has to actively participate in this process. And actually, the process of the cell needing to participate in the process has led my lab to recently discover the function of caspase 7. I'm going to tell you a little bit unpublished in-press data, caspase-7 is normally thought of as an apoptotic executioner, but it has never been known what its unique function is because caspase-3 accomplish apoptosis. And as I mentioned earlier, caspase-1 can cleave caspase-7, which doesn't really make sense because caspase-1 is not trying to activate apoptosis directly. But actually, the reason why caspase-1 activates caspase-7 is because it's important for this extrusion process. So here's a mouse that's been infected with salmonella. You can see a single extruding cells extrude singly, nice and cleanly. But without caspase-7, these cells extrude in these weird defective clusters. They can't get the job quite done, and then additional cells start falling out of the monolayer. And the reason for this is that caspase-7 is required to activate a membrane repair pathway. This is, and it's actually counteracting the gas German D pores so that the extruding cell can finish participating in the process of extrusion. The membrane repair pathway that it's activating is mediated by a protein called acid sphingomyelinase. This is a lysosomal protein that converts this lipid, which is a major constituent of the plasma membrane called sphingomyelin, into ceramide. Once this is converted, ceramide clusters, and because it doesn't have a head group, it spontaneously invaginates. What that does is normally, if there is a pore in a membrane, this causes a massive calcium influx, which stimulates lysosomal exocytosis. This has long been, been known, and this is been known for maybe a decade or so in membrane repair field. This releases a small amount of ceramide in the lysosome as well as the enzyme ASM onto the cell surface. Then ASM cleaves sphingomyelin at the surface to make ceramide, which endocytoses the pore. So if you have a small amount of pores, this basal membrane repair pathway can repair them. However, gas German D and caspase-1 is making an immense number of gas German D pores, which overcomes this basal repair process. So what happens is, in order to compensate for the number of gas German D pores that are being made, caspase-1 also activates caspase-7. Caspase-7 then leaves through the gas German D pore into the extracellular space where it meets ASM. Caspase-7 then cleaves ASM. And what happens is this cleaved ASM is hyperactive, and it now can make massive amounts of serum that can mediate the repair of many gas German pores. And caspase-7 and ASM need to keep up this membrane repair process for about 10 minutes. That's about how long it takes to extrude. So it's not like it has to go on forever. Eventually, gas German D is going to win, but this intestinal epithelial cell needs that 10-minute window to finish extruding before it loses all energetics and lyses through gas German D-mediated pore swelling and pyroptosis. So that's some data we're really excited about that's published going to be published pretty soon in Nature. All right. Coming back, you have noticed that gas German D is D. There are 6 members of this family. The 6th one may or may not be a gas German. It's probably going to be named gas German F at some point, ABF. Mice actually are kind of a strange outlier in mammals in that they've deleted B and have triplicated A and quadruplicated C. So many different gas Germans, and the mechanism by which these are activated has been, for most of them, actually recently discovered. They can all form the same pores. They all have that same 2-domain structure, a pore-forming domain and a regulatory domain, and a cleavage linker in the middle, but they're cleaved by different proteases. They also have very specific expression patterns. I'll get to the proteases in a second. The gas German A is expressed primarily in the skin, tongue, esophagus, stratified epithelium. multi-layer epithelial barriers. Gasdermin B also expressed in the uh, um, but also lymphocytes, stomach, liver, colon. Gasdermin C is primarily expressed in single columnar epithelium in the intestines primarily, but also in upper GI tract. Gasdermin D can be expressed in those same intestinal epithelial cells, but is very highly expressed in immune cells. Gasdermin E is expressed at low levels all over the place, is probably and is also inducible in its expression. And then Pejvakin, the sixth member, also has low expression in many different cell types. So what are all these? We know now mechanisms to activate 5 of those 6 gasdermins. So gasdermin D, cleaved by caspase 1 or 11. In the apoptotic pathway, normally caspase 8 and 9 activate caspase 3 to cause apoptosis. And granzyme B will also activate caspase 3 to cause apoptosis. If gasdermin E is expressed at sufficient levels, though, caspase 3 will cleave it and cause pyroptosis. So the hypothesis is that this is actually there's a couple different hypotheses as to how apoptosis and pyroptosis interface. One is that gasdermin E could be expressed only at certain times or only under certain conditions, which would essentially abort the apoptotic process to cause pyroptosis instead. Or gasdermin E may be expressed all the time at low levels, such that if the apoptotic processes go on for a long time without completing, you could abort to apoptosis. Or if apoptotic bodies remain present in the tissues for a long time without being cleared through erythrocytosis, which they should be cleared very rapidly, then this could cause secondary necrosis. It's still something that's being worked out as to why caspase 3 cleaves gasdermin E. Another, gasdermin B, was recently shown to be cleaved by a different granzyme. So natural killer cells and cytotoxic T cells deliver through perforin pores both granzyme B and granzyme A. So if the cell was expressing gasdermin D, then NK or CTL attack could cause pyroptosis instead of apoptosis. Cells toggle themselves between undergoing granzyme apoptosis or granzyme pyroptosis. Some cells express gasdermin B constitutively, but it also may be inducible in other cell types. So some cells, like intestinal epithelial cells, might be pre-programmed so that anytime they get attacked, they're going pyroptosis. Forget about apoptosis. And in this case, it seems that natural killer cells and cytotoxic T cells don't really know the difference because they deliver these two granzymes to all cells whenever they attack, is my understanding. But you could imagine that certain cytokines might change their expression profiles. Next, gasdermin C is said to be activated by caspase, was published to be activated by caspase 8, similar to gasdermin E. Like this would have the same kind of logic as to when this is expressed. And this is expressed, as I mentioned, constitutively in some cell types, like certain intestinal epithelial cells. So maybe whatever initiating pathway space 8 in those cells just want to go through a gasdermin mediated extrusion, probably in the case of an IEC, rather than a slower apoptotic extrusion pathway. And finally, and I think most recently, gasdermin A was recently published to be activated by a pathogen protease, the streptococcus protease called SPB, which is a protease that the bacteria uses for its virulence. This is a bacterium that infects the skin, and gasdermin A is primarily expressed in keratinocytes in the skin. And so this may be a way for the cell to detect that it has been compromised by the SPB streptococcal potentially invading cells and releasing SpB into the cytosol. With this paradigm, you would expect that many other pathogen proteases might also cause gasdermin A to activate, and it actually opens up room in these other gasdermins to add on little pieces of the linker that might be able to specifically detect the pathogen. And I think with that, I'm going to wrap up and see if we have any questions.
Thank you. Yes, thank you a lot.
That was a marathon.
Perfect. Excuse me?
That was a marathon of, that was a deluge.
Yes, but it's a great marathon. Yes.
It was awesome, Ed. I even gonna start with the questions while the students are, getting more confident to. I mean, the whole presentation was brilliant, and I really like your last data with caspase-7.
We're really excited.
It's beautiful. So I'd like to ask you whether you think caspase-7 also would function in apoptosis.
Oh, it does.
Provide it does to hold these other gasdermins to make pores. Not the caspase-7 as an executioner, but as this modulator of the appearance of gasdermin pores during apoptosis.
Yeah, we have not studied the other gasdermin pores during apoptosis, but actually in this same paper has data. We show that when natural killer cells and cytotoxic T cells attack with the perforin pore, that that pore also needs to be repaired. And we think that it needs to be repaired because apoptosis is a process that takes about a half an hour, requires membrane integrity, requires cellular energetics. Right. So we think, we show that during the classic example of listeria being cleared by cytotoxic T cell attack, which is a perforin-mediated clearance, you also need caspase-7 and you need this ASM activation in order to allow actually listeria to be cleared.
Wow.
That actually, I presented that data in Brazil at the Catarina's Keystone.
Yes, Keystone, yes.
So that presented maps to this exact same mechanism. So caspase-7 actually has one function, both in apoptosis and in inflammatory caspase signaling, which is to repair whatever pores might occur.
That's
nice.
Very cool.
Great.
That's very cool.
By at least some.
Yes.
That's pretty cool.
Yeah. Brilliant. Ed, maybe because of this complex interplay among these cell death pathways, many of papers that were published before the description of gasdermin D by using caspase-1 and 11 null mice could not be reproduced in gasdermin D mice. I'd like to know about your in vivo pyroptosis papers. If you did you revisit them by using gasdermin D null mice?
Yes. So all of the infectious models that we have, so we have some very strong phenotype caspase-1 mice, and all of them so far have been replicated with gasdermin D knockout mice. So in our models, the backup pathways are not functioning to clear the bacteria. It's a bit surprising to me, actually, that they're not. In vitro, we can replicate other labs' work that you see the backup pathways happening in vitro, and especially that the pathway that seems the most efficient to us actually is the caspase-1 to BID. to apoptosis pathway, it seems to happen pretty quickly. But we have, whenever we've looked at, so we look at bacterium called Chromobacterium, which has a very strong NLRC4 caspase-1 phenotype, and the Gasdermin D knockouts are nearly identical. There might be a little bit weaker. So there's room for that pathway, but it's not like solving the problem very a way that's really satisfying from an evolutionary perspective.
Yes, very interesting. Yes.
I don't doubt that the path
Guys,
is real, though. Yeah, student.
yeah.
Happy to take any questions from students. No, there's no such thing as dumb questions. Don't be shy. If you have
Don't be shy, guys. Yes,
Have-
Gustavo. Gustavo.
Gustavo Henrique, would you like to
Gustavo Henrique. Yes.
turn your microphone on? Or you prefer us to
Or you
read
prefer?
your question?
Yes. Okay, you can.
Shall I read
You
it?
can-
Oh, our Gasdermin D and
D and E detected.
our Gasdermin D and
Yes,
E
I am the library.
after cell death in the extracellular contents. Oh, can they also be detected in circulation, and could they be used to assess pyroptosis activation?
I think he's meant
I don't know if that's
biomarker.
Biomarker. Yeah, I don't know that that's. I'm not aware of people looking at that. I know they have. I think Eagle Ikelatz.
Bernardo Franklin. Yes.
Back biomarker of sepsis and that can be detected by staining and flow cytometry that the ASC has polymerized into this speck. I don't know if Gasdermin D and E are, I don't know if they're expressed at high enough levels to be a biomarker. HMGB1, I think, is a biomarker that investigators have used. We used it once to look at as a marker of pyroptosis in the blood. Gasdermin D, the N-terminus, actually has such affinity for membranes that it wouldn't be released as a soluble protein, but you could imagine the C-terminal regulatory domain should be soluble. Yeah, good question.
Because HMGB1 is not released only in pyroptotic cells.
Yeah.
It's also released under other necrotic cell death modalities.
Yeah, it's not specific.
Yeah.
I think
I have one more question. Oh, sorry.
Oh yeah, I was just saying that I think the best biomarker for pyroptosis is IL-1 beta, IL-18. So if you can detect that,
IL-1 beta can also be released by alive cells, not only by dead cells.
That's true.
So it is not a biomarker.
And there's also evidence that other pathways can cleave them. So caspase 8, I think there's some good evidence that it can cleave IL-1 beta and IL-18. Whether that's slower than caspase 1 or is actually as efficient, I'm not sure on the efficiency, but there's actually quite a bit of data that caspase 8 can cleave those cytokines too.
Yes. There is no specific marker for pyroptosis nor for necroptosis, and so on.
Yeah, especially from human samples, it's very, very, very challenging. In mice, we have it easier because we can use the knockout mice and then,
of course, yes,
make inferences from that.
So I have one more question regarding the best of course. I think my internet is quite unstable.
I think
yes.
You're back.
Are you hearing me?
Okay, now you're back. Yeah, now you're back.
Okay, so about the best inflammasomes. So you nicely shown the activation of NAPE and LRC4 inflammasomes by specific sensing of bacterial proteins by NAPE members. But there are increasing evidences about the activation of NAPE and LRC4 under other situations, non-bacterial infections, fungal, protozoan, and viral infections, and also under sterile conditions, especially in neuroinflammatory conditions. So I'd like to know your opinion about that, because for the majority of these situations, the NAPE, the role of NAPEs is unknown yet, is still unknown. I think it's only for HIV that they demonstrated the role of NAPE, human NAPE members sensing GP41 protein from HIV. I'd like to know your opinion about this. Do you think that NAPE could sense other proteins from these pathogens, or NAPE and RC4, rather than this, on the other hand, could be working as NLRP3 inflammasomes, sensing the cytosolic disturbances and so on?
Yeah, there's definitely room in the structure. Like if you think about that flagellin sitting on the nape and just like forming a bar, like there's a lot of room in that leucine-rich repeat for other binding domains. And the human nape, the single gene, it seems that the single gene can detect 3 different agonists that probably all have
more than 3
that they're like 2 alpha helices in the structure, but the sequences are not similar at all. So I think there's plenty of room for napes to bind, each nape to bind other agonists. In the mice, there may actually be additional napes that might be, I've never been clear actually whether NAPE3 is a pseudogene or is a real gene. So there might actually be more napes than flagellin and rod-centric people talk about. But also, if you think about NLRC4, it could act without the nape itself as well. It has the potential simply to bind an agonist and crank open it. One NLRC4 bound an agonist and opened, that would trigger the polymerization without the nape. So there's plenty of room in the system to add more agonists.
And there is a possibility that NLRC4 could act together with the NLRP3. There are some evidences in the literature.
Yeah. I've seen that as well. And then how they would interact together. How Wu would be a good one.
We don't know.
She would be the one to ask, like, what is the biophysics of those interfaces? Can they form mixed inflammasomes, or are we thinking about a multi-step signaling pathway, like like with Gasdermin?
Dual recruitment with the ASC doing a single platforms.
Yeah,
I think it
could enrich
more
multiple inflammasomes together.
Yes.
Or you could imagine, like if NLRC4 opened some Gasdermin D pores, you could be losing potassium, and NLRP3 could be responding to that. So there could be an amplifying multi-inflammasome step, kind of similar to the caspase 11 to NLRP3 non-canonical NLRP3 pathway. Anything that activates Gasdermin D should also run in that same pathway.
But it is intriguing that in these situations, we only see for NAPE and RC4 before than this NLRP3 activation. I think my internet's not.
Yeah,
Are you
it was
listening?
out.
Yes.
Now you're back
I
again.
am.
You're back. When this
Okay. So I was talking about even if innate NLRP4 inflammasomes are activated before the activation of NLRP3, we can see only one single spec, ASCII spec per cell. So it is intriguing to think about this. So we don't know if we can make two inflammasomes platforms in the same cell, or if these platforms are unified into a single spec one. So it is
I think I've seen in neutrophils that there are more commonly multiple specs. So there may be some cell type specific morphologies of the inflammasome polymerization pathway, which you could imagine occurring based on concentration of ASC, the diffusibility of the cytosol, how full the cytosol is of other organelles, and how rapidly they are synchronous through. Like if you have some kind of synchronous two NLRP3s activating in different parts of the cell, they could compete, make specs.
Yes.
So Ed, since you brought, I have a couple of things before we close. Just because you brought biophysics, a few times. I think I asked Karina at one point, but does anybody look at the NLRP3 activation by mechanical forces, by cytosolic elasticity, viscosity, or something like that? Because a lot of these, these signals also modify, the membrane. ATP that open up ion channels, or potassium, even these big clumps of stuff that crystals.
There was a paper many years ago that this isn't. I think you were asking about activation pathways, but there was a paper some years ago that talked about caspase-1 changing kind of like membrane or cellular turgidity, the turgor pressure of the cell and the
right.
cell
but
integrity.
that would be downstream. No,
Yeah,
I meant I meant because you also mentioned the role of the coadjuvant. I forgot the name, into the mitotic spindle. So when it's mitosis, then you don't have anything. And mitosis is something that really push the forces in the cell. So maybe there is something that the NLRP3 senses, mechanical forces to be activated.
I like that idea. Yeah, it's something really, which would kind of explain, if it was something very physical like that, it would explain why it's been so difficult for biologists to figure it out. We're all geneticists, biochemistry, cell biology. If it's really a physical thing. There's a recent paper on NLRP6 actually that talks about phase transition, and that it may be detecting and maybe monitoring phase transition from Hao also, which is a physical force.
Right.
So complicated but interesting topic.
Yeah, that would be good. Somebody that could look into it. And the other thing is just if you don't mind spending a little bit of time of saying the relationship between your view on pyroptosis and cancer, particularly in the tumor microenvironment.
I think there's a bunch of papers, or several papers recently that have been proposing that if we can switch apoptosis to pyroptosis in a tumor microenvironment, that you'll change the inflammatory state of the tissue, which will then alert the immune system to start attacking. And I think that's a great idea. I think there's some really promising data coming out on that.
But it's so controversial because I think it depends on the tumor tumor type. Because I think for the intestinal environment is good to have the pyroptosis, but for others is quite
some
controversial.
Tumors,
yes.
Some type on inflammation tumors like to be silent.
Yes.
Yeah, some tumors benefit from inflammation, others not. But also, I think even the same tumors will respond differently if the intensity of quality of the cells that are, being attracted to the tumor. So I think it's a very interesting room for studying apoptosis, pyroptosis in the tumor microenvironment.
Yes.
Okay, man, it was brilliant, really. Thank you so much.
Good.
A lot of material.
Thank you a lot.
Understand
Thank you a lot.
Students, and thanks for inviting me.
All right. Hope to see you soon in person.
See you soon.
Here in
Yes.
Brazil.
With the kids.
Yes.
All right.